Now students, second lesson in our textbook, that is sexual reproduction in flowering plants. Sexual reproduction in flowering plants. So before entering into this lesson, I would like to explain some of the important aspects of a flower. So in a flower, we'll see 4 whorls. That is, one is petals, sepals, then carpel, stamen. These are the 4 important parts of a flower. When these 4 parts are present, we'll call it as complete flower. Now, flower means it is a modified shoot. When bisexual flower comprises of androecium and then gynoecium. Androecium is nothing but the stamen. So if you come to the stamen, already in first PUC, morphology of flowering plants, there you came to know all the scientific terms. So stamen is male reproductive part of a flower. In this, we'll see anther is the part. Outwardly, if you see the anther, it is showing bilobed condition. So 2 lobes, united 2 lobes we'll see. That is called bilobed anther. Then second part of a stamen is long slender stalk is present, which is called filament. Long slender stalk is called filament. So anther and filament, these are the 2 parts of a stamen. So it represents male reproductive part of a flower. Then if you come to the gynoecium, so gynoecium is nothing but carpel. Another name is also there, pistil. Okay, gynoecium, it is female reproductive part of a flower. So if you come to the gynoecium, which is female reproductive part of a flower, it comprises of 3 parts. It comprises of 3 parts. What are they? 1, stigma, 2nd, style, and 3rd, ovary. In the previous class, reproduction in organisms, there I explained very clearly about the carpel or gynoecium, which is female reproductive part of a flower. So the 3 parts, 1 is stigma, 2nd is style, and 3rd, ovary. These 3 parts are present in a female reproductive part. Scientifically, it is said to be gynoecium. So, according to our content in the syllabus, first we would like to, or I would like to explain bilobed anther. It is showing 4 sides. That means you will see 4 sides when you take cross section. That is called tetragonal structure with 4 microsporangia. So you might be thinking, what is this 4 microsporangia? Microsporangia are said to be the pollen sacs. So the 4 microsporangia develops into pollen sacs, and the pollen sacs are packed with pollen grains. Is it clear, students? It is very important. Pollen grains only, the male gametes. Pollen grains are the male gametes. First of all, here, structure of microsporangium. So now we are going to learn what is the structure of microsporangium. According to this, we will see the section of this microsporangium. Here the diagram, please all of you look at the diagram. We can see here first epidermis. Outermost layer of microsporangium is epidermis. Next layer is, which is inside little bit, that is endothecium. And later here you will see middle layers, not layer, it is middle layers. So here epidermis, which is the outermost layer, endothecium, that is the second layer, and innerly if you see middle layers are there. These three, they are meant for protection and dehiscence of anther. So again I am repeating, these three layers, they are meant for protection and dehiscence of anther. That means, so when this dehiscence takes place means, here the stamen is having bilobed anther, right? So this anther, inside what we have seen, 4 microsporangia which develops into pollen sacs, and these pollen sacs packed with pollen grains. So these pollen grains, when they mature, then only what happens, this anther will break. So that anther, when it breaks, we'll see release of pollen. That is called dehiscence of anther. Now, innermost layer is tapetum. The innermost layer is tapetum. Here, this tapetum is nourishment of developing pollen grains. So, it is helping in the nourishment of developing pollen grains. So, very, very important competitive bit it is. For NEET or CET, these two are important. The 3 layers, epidermis, endothecium, middle layers, these 3 are meant for protection and dehiscence of anther. Whereas tapetum is nourishment of developing pollen grains. So this is also very important. Innermost layer, it is tapetum. Then if you see at the center, microspore mother cells are present. Microspore mother cells. So when we wanted to know about microspore mother cells, here sporogenous tissue, this is the very important terminology, because at the center of this only we'll see it occupies sporogenous tissue. So the center of microsporangium is with sporogenous tissue. So sporogenous tissue is very, very, very important, because from the sporogenous tissue only the microspore mother cell is produced. So microsporogenesis, another terminology, microsporogenesis. Microsporogenesis. So what is meant by microsporogenesis? The cells of the sporogenous tissue undergo meiotic division and form microspore tetrad. So 4 cells together we'll see, that is called microspore tetrad. Each cell of sporogenous tissue capable of giving rise to microspore tetrad. So as we know here, microsporogenesis means the cells of the sporogenous tissue. Where is this sporogenous tissue? The sporogenous tissue is occupies the center of microsporangium. So that each cell of that is capable of giving rise to microspore tetrad. Is it clear, students? So microspore tetrad is important. Then definition for microsporogenesis. Formation of microspores from pollen mother cell is called microsporogenesis. Again I am repeating, formation of microspores from pollen mother cell is called microsporogenesis. So this is about the structure of microsporangium. Then coming to the another part, that is microspores. Just now we learned microspores. Microspores in cluster of 4 cells. That means microspore tetrad means it is with 4 cells. Microspore tetrad, isn't it? So the anther mature and dehydrate the microspore, then dissociate and develops into pollen grains. Again I am repeating, microspores are there, no? How they are? They are with 4 cells together. That is called microspore tetrad, cluster of 4 cells. Microspore tetrad, isn't it? Then the anther mature and dehydrate the microspore, then dissociate and develop into pollen grains. By the dehiscence of anther, pollen grains are released. Already I told to you here, pollen grains are present or they are packed inside the pollen sacs. So pollen sacs are nothing but the 4 microsporangia developed into pollen sacs. So in the first part of structure of microsporangium, here we came to know these 3 layers are responsible for protection and dehiscence of anther. So I have given very clear-cut information about the dehiscence of anther. That is only here. Once again, I am telling that by the dehiscence of anther, pollen grains released. Now, the bilobed anther, now it breaks. When it breaks, the pollen will be released. Pollen grain represent male gametophyte. Already I told to you. Now students, pollen grain. So pollen grain is male gametophyte. Pollen grain is considered as male gametophyte. So here we have seen, this is the structure which is called tetrad cell, having 1, 2, 3, 4. 4 cells collectively. So here the pollen grain tetrad we are seeing, and then when it develops we can see here, this is the pollen grain actual structure. So pollen grains are developed from microsporangia, 4 microsporangia. Pollen sacs are packed. Inside the pollen sacs we will see pollen grains are packed. So this is the structure of pollen grain, and here the outer layer is called exine. Exine is the outer layer. Inner layer is called intine. And then if you see in this pollen grain, it is having 2 cells. One is vegetative. So, exine made with sporopollenin. As I told you, exine is the outermost layer of the pollen grain. So, this is the outermost layer. It is made with sporopollenin. Sporopollenin is very important for NEET exam. So, exine is made with sporopollenin. This sporopollenin will have importance. What is that importance means? Exine is resistant to strong acids and high temperature, also alkali. So resistant to strong acids, high temperature and alkali. That is the exine importance. That means because it is made with sporopollenin. So because of this substance only, it is having resistance to strong acids, high temperature, alkali. Then coming to the another point in hexine, hexine has prominent apertures. So for example, they have prominent apertures means you will see like this. So each it is having prominent apertures like this. So hexine is having prominent apertures, and what they called the prominent apertures are said to be germ pores. The prominent apertures are said to be germ pores, where sporopollenin is absent. This is most important for NEET, because wherever the germ pores are present, these are the germ pores. The entire pollen grain comprises of, that means the hexine only comprises of many prominent apertures. They only called germ pores, where means at the germ pores region sporopollenin is absent. This is very important for NEET exam. Then let us come to intine. Inner layer of pollen grain is called intine. Intine made of cellulose and pectin. This is also important question. Intine made of cellulose and pectin, whereas hexine is made with sporopollenin. Then intine made of cellulose and pectin, I told no. Pollen grain contains two cells. Here in this diagram we are seeing the two cells. This is one cell and this is another cell. So the cell which is bigger in size, that is called vegetative cell. The cell which is bigger in size, it is called vegetative cell. The cell which is small in size, it is called generative cell. So this condition of pollen grain is said to be two-celled stage. It is called two-celled stage. Why it is called two-celled stage? Because it comprises of vegetative cell and generative cell only. So two-celled condition. According to the scientists, what they are telling is at this two-celled stage only the pollen grain is shed off. That means the anther breaks, dehiscence is taking place. Later the pollen will be released, like that scientists are telling. Let it be. Then here let us examine or let us see the structures of these two. Vegetative cell how it is and generative cell how it is. So vegetative cell here, as I told you, it is bigger in size or bigger cell. Then generative cell is small cell or smaller in size. Second point under vegetative cell is irregular shaped nucleus is present. So irregular shaped nucleus we can see at the vegetative cell. The nucleus is not completely spherical. It is irregular in shape. Then if you come to the generative cell, it floats in the cytoplasm of vegetative cell. That is the important point. That means because of its small size of generative cell, because of high amount of cytoplasm which is present in vegetative cell, so that's why this cell which is called generative cell, it is floats in the cytoplasm of the vegetative cell. So that's why here what we have to observe is we should know vegetative cell how it is and generative cell how it is. And the vegetative cell is having more amount of cytoplasm. That is also one of the important point. For that sake, the small cell which is generative cell, it floats in the cytoplasm of vegetative cell. Now next let us come to the three-celled state. Three-celled state means or stage means generative cell divides mitotically and gives two male gametes. Here I mentioned very clearly three-celled stage. Already I told two-celled stage. Now I am telling three-celled stage. What is a two-celled stage? The two-celled stage means the pollen grain comprises of large vegetative cell and small generative cell. That condition tells that it is in two-celled condition. Then three-celled condition means this so-called generative cell, what happens? This will divide mitotically. This is most important. Mitotically and gives rise to two male gametes. And these two male gametes, they are coming into the pollen tube. They will enter into the pollen tube. Then later what happens, the inner wall which is called intine, made of cellulose and pectin, I told no. Later what happens here, the vegetative cell and generative cell, when they are present, we'll call it as a two-celled stage. The generative cell divides mitotically and gives two male gametes before pollen grain shed. Then this is called three-celled stage. Hope you understood. So here why it is called three-celled? Generative cell is one cell, and when the nucleus of that generative cell divides mitotically, then we will see as a result of that, two male gametes also produced. So according to that, we can say three-celled stage it is. So generative cell, when it undergoes mitotically, that means the nucleus undergoes division mitotically, then it gives rise to two male gametes. That is called three-celled stage. And further explanation, you please listen carefully. Please lend your ears. Some pollen grains are harmful. So some are very harmful to the human body, and not only that, for animals also. For example, Parthenium. Parthenium is there. It is said to be carrot grass. The other name for Parthenium is carrot grass. So it causes allergy. So what type of allergy it is? Bronchial asthma. So bronchial asthma we'll see in the case of some of the pollen grains. So whenever the pollen grains, that means the Parthenium plant pollen grains are very minute. When we are passing through the plants, we don't know the air is with these minute pollen grains of Parthenium. When we inhale, some of the person's immunity, when they are not having good immune power, that such a type of persons are proneable to that bronchial asthma. So it is a allergic disease. Understand? So for that sake here, they have given in our textbook, they have given pollen grains are harmful, and for that example is Parthenium, that is carrot grass, cause allergy. That allergy is bronchial asthma disorder, and chronic respiratory problems also will be seen in such persons. Next is pollen grains are rich in nutrients. So pollen grains are rich in nutrients. Pollen consumption claimed to increase the performance of athletes and race horses also. So here pollen grains are harmful. At the same time, some pollen grains which are rich in nutritive, that means they are very rich in nutrition condition. That's why some of the athletes, they are using the pollen grains, and for that sake they will be getting energy, and at the same time race horses also. Then in the case of rice and wheat, please, if you have notebooks, you can make a note. Rice and wheat, these are the two examples. Pollen grains lose its viability within 30 minutes. I am repeating, important, competitive point of view important. In rice and wheat, pollen grain lose its viability within 30 minutes of release on stigma. So what is viability? Viability means it is ability to survive or ability to live. That is called viability. If you don't know the meaning, please make a note. Viability means it is ability to survive or live. Okay. So here what is happening? The rice and wheat within 30 minutes only. So the viability is their survival time is 30 minutes only. So after the release on stigma. So all these pollen grains. Next pollen grain explanation, what we'll see, pollination we'll see. So in the case of pollination, what is the definition for pollination? Transfer of pollen grain from the anther to the stigma. Okay, transfer of pollen grain from the anther to the stigma. That is called pollination. So pollination, for pollination purpose, many agencies are present. That means pollination by wind, pollination by water, pollination by insects, pollination by so and so. Understand? That means animals also, birds also. Okay. So all these are helping in pollination. And the best pollinators are honeybees. Nowadays, when we are watching our TV, what we are listening means the scientists, they are telling the honeybees are going to be extinct. So that is also one of the big problem, because without pollination we won't get any vegetables or fruits. Okay students, so once again you please listen carefully. The viability of rice and wheat is 30 minutes only. Then Rosaceae. So here three families are given in our textbook. One is Rosaceae, second one is Leguminaceae, third one is Solanaceae. So Rosaceae, Leguminaceae and Solanaceae, in that the pollen viability is for months. They can survive months, not 30 minutes. Okay. In the case of rice, very few minutes only, 30 minutes. But in the case of, so it varies. Rosaceae, Leguminaceae and Solanaceae pollen grains viability is for months. And one more important point is, which is important for competitive also, pollen grains are stored in liquid nitrogen. So how we are keeping our money in banks, like that the pollen grains also preserved. So how they preserved means these grains are stored in liquid nitrogen at minus 196 degrees of centigrade. Very, very important. What is important here? They are stored, pollen grains are stored in liquid nitrogen at what temperature? Minus 196 degrees of temperature. Such pollen are kept in seed banks. They are kept in seed banks and used in crop breeding programs. So the farmers, they are using in their cultivation. Then here one more important point, what I would like to say is, pollen grain measures about 25 to 30 micrometers. Please make a note. Pollen grains measuring about 25 to 50 micrometers in diameter. So that is also one of the important point. So that's why students here, once again I would like to recap the pollen grain, which is considered as male gametophyte, and then the pollen grain comprises of, how this pollen grain is coming. This is a microspore tetrad. How this microspore tetrad is coming? From Microspore mother cell. So the microspore mother cell, from which part it is coming? Genus tissue. Sporogenous tissue is there, no? So each cell is capable of giving rise to microspore tetrad. Okay, students? So that's why here it is in tetrad condition, and the pollen grain, which is considered as male gametophyte, and this is the structure of pollen grain. The outermost layer is exine and the inner layer is called intine. Exine is made of sporopollenin. This is important for NEET, and it is resistance. Because of this substance, it is resistance to strong acids, high temperature and alkali. Exine has prominent apertures also. So these are prominent apertures which are present on exine. So these apertures are called germ pores. Germ pores. So wherever the germ pores are present, there is no sporopollenin. So sporopollenin is absent. This is important for NEET exam. Then if you come to the intine, intine is made of cellulose and pectin, and pollen grain contains two cells. So which are the two cells? Vegetative cell and generative cell. Then mostly in two-celled condition only, pollen grain will shed, like that scientists are telling. What is three-celled stage? Three-celled stage means generative cell divides mitotically and gives two male gametes. This is called three-celled stage. Okay, students, do you understand? Next we'll see. Next, students, next we'll see, as I told you, the female sexual part of a flower. This is called carpel or pistil. So here, according to the number of carpels, monocarpellary, that is single carpel is present, or single pistil is present. Such type of a flower is said to be monocarpellary. So mono means one, single carpel. Example, coconut, mango. These two comes under monocarpellary. Then coming to the second terminology, multicarpellary. More than one pistil. More than one pistil, if it is present, it is called multicarpellary. Okay, students? Then for this, example is papaver. So examples also you should remember, in competitive exam they used to ask. Monocarpellary, single, presence of single pistil. Example, coconut, mango. Then multicarpellary, more than one. Example, papaver. Then coming to the syncarpus. So syncarpus means when there are more than one pistils may be fused together. So if you make our fingers fuse together, like that, the pistils will be in fused condition. So this fused nature of pistils is called syncarpus. And here one more importance is multicarpellary syncarpus. Example is papaver. Again I am repeating, multicarpellary syncarpus. What is syncarpus? When there are more than one pistils may be fused together. So here fused. When they fused, it is called syncarpus. Yes, syncarpus, when there are more than one pistils may be fused together, that is called syncarpus. So fused together. So when the pistils are fused together, that is called syncarpus. Next, apocarpus. Pistils are free. When pistils are free, they are not in fused condition. Such a type is said to be, that means example for that is mycelia, and it is called apocarpus. So students, you should know syncarpus condition and apocarpus condition. Syncarpus is fused condition, whereas the pistils are fused, and in apocarpus pistils are free. Example for this is mycelia. So multicarpellary apocarpus example is mycelia, and multicarpellary syncarpus is example is papaver. Then monocarpellary, here already I said coconut and mango. Then let us see placenta. What is placenta? So in the case of placenta, here we are seeing this is the entire portion which is called carpel or pistil. This is the female reproductive part of a flower, isn't it? So it comprises of three parts. One is stigma, second is style, and this is the ovary part. Stigma, style and ovary is there, and then below the ovary there is a thickened portion is present at the end of the ovary, and that is called thalamus. So stigma, style and ovary, in this is the ovary part. So inside the ovary, you people should remember carefully, inside the ovary you will see mass of tissue in the ovarian cavity. So this inside cavity of the ovary is called ovarian cavity. So it is filled with a mass of tissue, and that is called placenta. Then locules, one more terminology, locules. Locules means they are small chambers. The small chambers which are present on the placenta, they are called locules. Then according to the number of locules, the name is given. If it is only one locule in the placenta, then it is called monolocular. Then if there are two locules, bilocular. 3 locules, trilocular. 4 locules, tetra. Like that it goes on. Then ovule. What is ovule? Inside the locules. So locules means they are the small chambers present on placenta. So those small chambers inside, we'll see ovules are located. Okay. So they are called ovules. And one ovule we'll see in an ovary. Example, wheat, paddy and mango. In these three, we'll see only one ovule in an ovary. Okay. Then many ovules. So we'll see many ovules in the case of papaya, watermelon and orchids. Again, papaya, watermelon, orchids, best examples for many ovules. Now students, here as I told you, placenta, already in first PUC we come across the placenta according to the morphology of flowering plants. Their types of placenta we came to know, isn't it? So then here locules, ovules, all these detailed description in this chapter only given. So here locules are the small chambers inside the locules. Ovules are present. I said ovule will be situated in a locule. So ovules will become into seeds. That part here I didn't write, but it is your duty to remember. Ovules will become seeds. Ovary will become, the ovary, this part is becoming fruit. When it becomes fruit, when it is a fertilized one or ripened ovary, that will become into a fruit. Whereas ovules, they will become into seeds. Okay. After the fertilization, that means pollination is over. Then in the case of angiosperms, we will see double fertilization. Yes, no? So in that double fertilization, the embryo, after the fertilization, embryo is formed. That the embryo will become into, that means zygote will become into embryo. After the fertilization, as a result, we will see zygote. That zygote only will become into embryo. Okay. And embryo is, the development is inside the ovule only. So in our chapter we will see the anatropous ovule. So in the next class what we have to see is the structure of anatropous ovule, and one more important point here is the ovule is the common name for megasporangium. So megasporangium is the scientific term, and for that commonly it is named as ovule. Okay students, so here as I told you, ovules will become into seeds, ovary will become into fruit, and the wall of the ovary only the fruit wall. That means if you take any fruit, the fruit external layer is called pericarp. Don't forget all these scientific terms. Pericarp, P-E-R-I-C-A-R-P. Let us see the megasporangium, which is called ovule. So in the case of megasporangium, which is called ovule, in our syllabus the ovule which is called the LS of anatropous ovule is given. So let us see the structure of anatropous ovule. So first of all, the ovule is a small structure attached to the placenta. So this ovule is attached to the placenta by a funicule. This is a stalk-like structure which is called funicule. So it is attached to the placenta. The body of the ovule fuses with the funicule. So this entire ovule body is fuses with the funicule in the region called hilum. So this hilum is the part which is said to be the body of the entire ovule fuses with the funicule, and that fuses region is said to be the fusing region is said to be the hilum. The junction between ovule and funicule. Now the junction between ovule and funicule is hilum only. So I am telling two important sentences here. Hilum is the one, the body of the entire ovule fuses with the funicule. That region is called hilum. And another sentence is the junction between ovule and funicule is hilum only. Okay students, then ovule has two protective layers. So you can see two protective layers here. Outer integument, two integuments we can say, they are the protective layers. Outer integument is there, inner integument is there. So usually what happens, the outer integument will be the outer layer of the seed, which is called testa. As I told you, ovules will become into seeds, isn't it? So in first PUC, morphology of flowering plants, there just to the end of the topic, we came to know seed structure. So seed is having outer coat which is called testa and inner coat which is called tegmen. So outer integument will become into testa of the seed and inner integument will become into tegmen of the inner coat of the seed. So this is very important competitive bit. Then these integuments encircling the nucleus tissue. So here you will see nucleus tissue is present. These are the integuments, and inside you will see this entire portion is with the nucleus tissue. So nucleus tissue is a nourishing tissue for the ovule. Inside the nucleus tissue we will see embryo sac. This is called embryo sac. So nucleus tissue is the one which is a nourishing tissue. The nourishing tissue inside we will see embryo sac. In this embryo sac we will see the cells which are said to be these two are synergids and the middle two are polar nuclei. And here at the Chalaja region, these three cells are said to be antipodal cells. So this spacious portion of the ovule, it is called Chalaja. Then according to the different flowers, and the ovules will vary from flower to flower. But here one more thing which you have to remember is the embryo sac is at the center of the ovule. Embryo sac is at the center of the ovule. And in the ovule we will see only one single opening portion. So this is the single opening portion which is called micropyle. So micropyle is the opening portion of the ovule. So it is opposite to Chalaja. So Chalaja opposite, micropyle opening is present. This is the opening. So what is the use of this micropyle means? Through this micropyle only the pollen tube will enter. So here in between these two synergids, certainly here there is a egg cell is present here. So egg cell is there. Don't forget about that. In between these two synergids, egg cell is present. At the center of the embryo sac, two polar nuclei are present. And at the anterior portion of the embryo sac, three antipodal cells are present. So from the micropyle only, the pollen tube will enter into the egg cell. So before entering, what happens? Here finger-like projections, please observe students, here finger-like projections are there. They are nothing but cytoplasmic projections only, and those finger-like projections are said to be filiform apparatus. Very, very important filiform apparatus for NEET exam, because this filiform apparatus only will guide the pollen tube where to enter and where not to enter. So it only will guide the filiform apparatus. When the pollen tube enters into the embryo sac, at that time first of all the filiform apparatus will make that pollen tube to enter into the synergid, one of the synergid. Then immediately what happens? The pollen tube is carrying two male gametes. We are knowing already. Two male gametes. How the two male gametes are produced? The generative cell nucleus, when it divides mitotically, then as a result two male gametes are produced. So those two male gametes are inside the pollen tube. That's why through pollen tube, when the male gamete, one of the male gamete will enter into the one of the synergid, and the remaining one, what happens? It will disintegrates, and that one of the male gamete fuses with the egg later. Then the fertilization has taken place. So that is called zygote only. So the zygote will become into embryo. So this is the detailed explanation of anatropous ovule. And one more important thing I would like to quote here is, as I said you, outer integument will become into outer coat of seed testa. Inner integument of the ovule will become into inner coat, which is said to be the tegmen of seed. Thank you, students.